Hello students, good morning. I hope I am audible to you. चलो भाई चिर Yes, we were discussing about one-day motion. So we discussed about acceleration and what will be the distance traveled in 1 second, right? So the velocity is 100 meter per second and acceleration is plus 2 meter per second square. Acceleration is plus 2 meter per second square and velocity is 100 meter per second. What will be the velocity in next 1 second? In next 1 second, what is the velocity? And again in the next 1 second. What will be the velocities? This is 100 meter per second. So in the next 1 second, since acceleration is positive, this value will increase by 102 meter per second, right? Then it will increase by 2. So speed is 104. Yes, what is the distance now? What is the distance traveled? It is simply velocity plus the value of a by 2. What is the value of a by 2? What is the value of a by 2 here? A by 2 is 1, right? So just add a by 2 the given velocity. So this will be 101, right? 101 meter. And what is the distance traveled in the next interval? This is 101 plus 2, right? 101 plus 2, this is 103. So distance traveled in first interval will increase by the value of a . So we discussed this in the last lecture. So we shall discuss another concept, which is a bullet. A bullet through a block. Bullet through a block. Okay. We have a wooden block which is fixed here, and there is a bullet passing through the block. Okay. There is a bullet passing through the block. We have a bullet whose velocity is u, and from this point to this point, the velocity will not change. Okay. From A to B, from A to B, the velocity will remain constant. Now, what happens when bullet enters the block? From A to B, the velocity is constant. What happens when the bullet enters the block? This is a wooden block. This is a wooden block. What happens when bullet enters if there is a bullet here? What happens when it enters the block? Oh, Piroliran. Comment over huh? Good morning, everyone. The speed will decrease, right? When the body or the bullet enters the wooden block, the speed will decrease inside the block, right? So there is a possibility that the bullet comes to rest, right? There is a possibility that the bullet comes to rest inside the block, right? So d is the distance here, and what is the distance traveled by bullet before coming to rest? We have to calculate that. Okay. Okay, speed ಯಾಕೆ ಕಮ್ಮಿ ಆಗುತ್ತೆ ಅಂತ ಕೇಳಿದರೆ, why the speed decreases? Because this block will resist this, right? When a bullet enters a block, wooden block, it's a common sense that the speed will decrease. Why? Because the acceleration is negative. After some distance, the bullet comes to rest. What is the distance here? I will take this as distance traveled by bullet. Okay. So what is the distance traveled by bullet? We can calculate this. We can use this formula: v square is equal to u square plus 2as. What is the final velocity? Final velocity is velocity at c. What is the initial velocity? It is the velocity with which it enters. So here velocity of bullet is u, and at this point also the velocity will be u itself. So initial velocity is u. So velocity at c is 0. This is u square plus 2. What will be the acceleration? Acceleration will be negative. And what is s? S is the distance traveled by bullet. So if you solve this, it is minus u square is equal to minus 2a distance traveled by bullet. So minus minus cancel. So what is the distance traveled by bullet? Is equal to u square by 2a. Distance traveled by bullet will be u square by 2a. So this is distance traveled by bullet. We can name in this way. Okay, so this is a simple concept now. we can change the concept here. Sometimes the bullet comes out of the block, okay. Sometimes the bullet does not comes to rest, but it will emerge out from the block. The bullet will comes out of the block depending on the retardation, okay. So it may come out with velocity v. It will enter with velocity u, and the bullet may not come to rest within the block. Inside the block, it does not come to rest. It will come out of the block with the speed v, okay. So what should be the acceleration? What should be the acceleration so that the bullet will come out, okay, or we can call it as maximum retardation. What is the maximum retardation given by the block so that the block emerge out? So we can solve this. What should be the maximum acceleration? So v square is equal to u square plus 2as. We can use this formula. So if bullet comes here, if bullet comes out and the velocity is 0, the bullet travels the block completely, and when it reaches here, its velocity is 0, but this will comes out. Now what should be the maximum acceleration? So we can use this. 0 is equal to u square plus 2a, and this is minus here. What is the distance traveled here? Distance traveled is the thickness of the block. If you solve this, this will be a max is equal to u square divided by 2d. A max is equal to u square by 2d. If the acceleration is greater than a max, or we can tell if acceleration or retardation is greater than u square by 2d, what happens? Bullet does not come out of block. Right. If this condition is satisfied, if acceleration is greater than u square by 2d, the bullet will not come out of the block. Okay. So to come out, for a bullet to come out of the wooden block, the acceleration should be equal to this, or it should be less. Okay. What is the acceleration to come out? Acceleration should be less than or equal to a max. This is one of the condition. Retardation given by the block should be less than a max value. This is one of the condition. Another condition is that if a velocity is very large, here if the velocity is very large, then it can cross the block completely, right? So it depends on retardation also, it depends on velocity also. Okay. Velocity of bullet. So we have a bullet moving in a path, and we have a block here, wooden block. So I have thickness of the block as 20 centimeter. Thickness of the block is 20 centimeter. Okay, so the bullet will enter. The bullet will enter with a speed of 5 meter per second. Check whether and acceleration is 0.5 meter per second square. Acceleration of the block is 0.5 meter per second square, and thickness is 20 centimeter, and bullet is entering the block with speed of 5 meter per second. Check whether bullet comes out. Check whether bullet comes out of the block. Let us check whether the bullet comes out of the block. You have to check whether the bullet comes out of the block or not. Okay. Or we can use one more method. So we can use this v square is equal to u square plus 2as take v outside. Take v is equal to 0. What is initial velocity? Initial velocity is 5 meter per second square. And 2. What is the retardation? What is the retardation here? Retardation is 0.5. So this is negative. This is the retardation value. So what is the distance traveled? You just calculate here. This is the second method. Okay. So if you calculate the distance here, this is 5 square. When it comes here, it is 25 divided by this is 2 into 0.5 is 1. The distance traveled is 25 meter. The distance traveled is 25 meter. So the distance traveled is greater than the given distance, right? The distance traveled by the bullet is greater than the given distance. So definitely it will pass through the block. Or one more method, we can use this method. We have to calculate the Amax value. We have to calculate the Amax value. How to calculate Amax? It is u square divided by 2 into thickness of the block. A max is equal to u square by 2t. Let us calculate A max value. A max is, what is u square? U square is 5 square then 2. What is the thickness? Thickness here it is 20 centimeter. It is 0.2. 20 centimeter should be taken in SI unit, so which is 0.2 meter. Now we can simplify this. What happens to the A max value? A max is equal to 5 square is 25 divided by, what is 2 into 0.2? 2 into 0.2 is 0.4. If you solve this, what is the answer for here? A max will be, if you solve this, this is 25 divided by 4 by 10. It goes to the numerator. So A max is equal to 250 by 4. So what is A max? A max is equal to 250 by 4. So this is 125 by 2. 125 by 2 is nearly 62.5. So here see, here the A max value is 62.5, and what is the value of A? A value is 0.5. So A value is less than A max. A value is less than A max, so definitely the bullet will come out. Okay. Since the retardation given by the block is less than A max, it will come out. Okay. So one more question they may ask from this concept. Since bullet come out of the block, with what speed does it come out? They may ask this question. With what speed this may come out? Okay. With what speed does it emerge out? So we already know the bullet will come out of the block, but with what speed? To calculate this, we can use this formula. V square is equal to U square plus 2AS. V is the speed with which it comes out. U is the speed with which it is entering. So it is 5 square and plus 2. What is acceleration? Acceleration is minus 0.5. And what is the distance traveled here? Since this comes out of the block, the total distance traveled by the block is 20 centimeter, which is 0.2. This is 25 plus 25. This is plus into minus minus. So this is 2 into 0.5, it is 1. So 1 into 0.2, it is 0.2. So this is V square. What is the value of V? V is root of 25 minus 0.2, which is 24.8. So what is the root of 24.8? It is 4.97, right? This is 4.97 meter per second. So the block will come out and its speed is 4.97, right? I hope this concept is clear. What happens when a bullet enters the block? The speed will decrease. How to calculate the final speed? First you have to check whether the block will comes out of the block. If the retardation given by the block is less than max, then it will come out. Then if it comes out of the block, what is the velocity? You can calculate by using this formula. In the place of distance, you have to take the thickness of the block. Okay, now there is a second type of question in this concept. There is a block here. There is a block. And if a bullet enters with speed u, its speed becomes u by n. Okay. If a bullet enters a block of thickness t, if initial speed is u, after coming out of the block, its speed becomes u by n. Okay. There is a single block whose thickness is t, and when a bullet enters the plank, it will come out. What will be the speed? The speed is u by n. Okay. Here speed is decreasing. Now, how much planks are required to stop bullet? This thing we have to calculate. Okay, here n is a number. Suppose here speed is 10, and if it comes out, what is the value of n here? Here the speed is 10 meter per second, and when the bullet comes out, the velocity is 5. By what factor it is decreasing? You can see here. This is 10 and this is 5. Usually this can be written as 10 by 2, right? Here n is the factor by which speed will decrease. What is n? N is a factor by which speed is decreasing. You can look at here. Here the speed is 10. When it comes out, the speed is 5. So the 5 can be written as 10 by 2, right? So what is the n value? N value is 2. What is n? N is the factor by which speed will decrease. Okay. Whether the speed has become half. If speed has become half, then n value is 2. If the speed is 2.5, speed is 10 and final speed is 2.5. So by what factor did it decreased? So whether it is whether it has become half or whether it has become 1 by 4 times. So this 2.5 can be written as 10 by 4, right? N is always 2 or not, sir? No, n can be anything. If speed becomes half, then n is 2. If speed becomes 1 by 4, in this case the speed has become 1 by 4, right? Initially the speed is 10, then it became 2.5. The initial speed has decreased by the factor 4, right? So here n value is 4. Sometimes it becomes 1. Suppose the final speed is 1 meter per second. What is the factor here, or what is the value of n here? Bullet enters the block with 10 meter per second and it comes out with 1 meter per second. Yes, the factor depends on final velocity and initial velocity. So what is n value? Here the speed has decreased from 10, it becomes 1. This speed is 1. By what factor did it decrease? What is the n value here? This 1 can be written as 10 by 10, right? So initial speed is 10. So this is 10 by 10. So what is the value of n? N value is n value is also 10. Oh, how to calculate this n value? This is simple. How to calculate this n value? N value is calculated by initial speed by final speed. Okay. N is the ratio of initial speed to final speed. If this is 10 and this is 5, what is n value? 10 by 5 is 2. So n value will be 2, right? Okay. So now look at here. Suppose if you use one Planck, initial speed is u and the final speed is u by n, then how we can stop a bullet? If you use one Planck, the speed is decreasing. If initial speed is u, the final speed is u by n. Okay. Now, how we can stop a bullet? Because if we use one Planck, the bullet does not stop. What we can do is, we have to increase the number of Plancks, right? If you use one Planck, the bullet does not stop. How to stop the bullet completely? We have to increase the number of Plancks. If you use many Plancks, the bullet may stop, right? From one Planck, the bullet did not stop. What you have to do? You have to use n number of Plancks. So then, after traveling some distance, the bullet may come to rest. Okay. From one Planck, the speed does not decreases. Then, if you use n number of Planck, the bullet may stop. Now, in this case, or from this diagram, how many Plancks are used here? From this diagram, how many Plancks are used? 1, 2, 3 and 4, right? 4 Plancks are used here. So we have to calculate this number of Plancks required, and all Plancks are identical. All Plancks have same thickness. All Plancks have same thickness. So how much Plancks are required to stop the bullet? So how to calculate number of Plancks required? Okay. We shall see how many Plancks are required. So first equation is that we can use this. Initial velocity is v, final velocity is initial velocity is u, final velocity is v. V square is equal to u square plus 2a. Yes, we can use this formula. Here, what is the final velocity? Let us apply this equation for one Planck. What is the final velocity? Final velocity is u by n whole square. It is equal to, what is initial velocity? Initial velocity is u square. What about acceleration? Acceleration is negative. So 2a. What is the distance traveled? Distance traveled is t. Right? For one Planck, what is the distance traveled? T. For one Planck, what is the initial velocity? U square. What is the final velocity after coming out? If you take one Planck, the bullet enters with speed u and it will come out with speed u by n. So what is the answer for this? This is u square by n square, which is equal to u square minus 2at. Okay, let us solve this. If you solve this, what is the answer? this can be written as u square divided by n square and minus u square is equal to minus 2at. Right. So what is the acceleration here? If you solve this, this is u square minus n u square divided by n square is equal to minus 2at. And let us simplify this. U square is common. U square is common, then this will be 1 minus n square divided by n square, which is equal to minus 2at. If you take 2 and t to the right side, what happens? So minus a value will be u square by 2t into 1 minus n square by n square, right? So what is the value of minus a? Minus a value is u square by 2t, 1 minus n square by n square. Okay, now we shall simplify this, and now we will apply this equation to n number of planks. So if bullet enters with velocity u and it stops, if bullet comes to rest, bullet enters with speed u and it comes to rest, what is the distance traveled? How you can calculate? It is calculated using what is the distance of one plank, or what is the thickness of one plank? Thickness of one plank is t. If you use n number of planks, then it distance traveled is n into t. Okay. What is the distance traveled by the plank after it come to rest? It is n into t. What is n? What is n? N is number of plank. Capital N is number of planks. Capital N is number of planks. Okay. If you use 3 planks, if you use 3 plank, if bullet comes and it stops, and what's the distance traveled by the plank? It can be written as n into t. Okay. Here there are 3 planks, then I can take 3t. Okay. If it takes 3 planks, what is the distance traveled? It is 3 into t. So since it is taking n number of plank, what is the distance traveled by bullet? It is n into t. So let us apply the data to the formula. V square is equal to u square plus 2as. What is the final velocity? If bullet come to rest, final velocity is 0. What is initial velocity? Initial velocity is u square plus, here acceleration is negative, right? Acceleration is negative. So minus 2. What is value of this can be written as minus 2as. So 0 is equal to u square. What is the value of minus a? Minus a has been calculated here. Value of minus a is plus u square divided by 2t, 1 minus n square divided by n square into s. Right. I have substituted the value of a, and what is the s here? S is the distance traveled by the bullet. It is n into t. So t and t get cancelled. T and t get cancelled. So we have to simplify this. So u square goes to the left side. This is minus u square, and this will be u square by 2 This is 1 minus n square. What we can do here is, I can shift this can be written as n square minus 1 divided by n square, but you have to take minus outside, okay. Is it clear? Here the term was 1 minus n square. I have written as n square minus 1, I can take minus outside. If there is a term a minus b, this can be written as b minus a, right? But we have to write a minus outside. Okay, we have 2 here. The 2 get cancelled here, so I can leave this, and we have n here, right? The 2 and 2 get cancelled here, and t get cancelled, so I can cancel minus and minus and simplify this. If you simplify this, this u square and u square get cancelled, so we are left with 1. You can take n square on the right side, so this is n square divided by n square minus 1, it is equal to n, right? You will get this formula, n square divided by n square minus 1. So number of plank required is n square divided by n square minus 1. Yes, you can also calculate minus 2a also, simple thing. You can solve the equation in any method, but it should be correct. Right, so we shall solve a problem now. There is a plank of thickness 10 centimeter. Initial speed of the bullet is 5, and it comes out with speed 2.5 meter per second. Okay. The bullet enters with speed 5 and comes out with 2.5 meter per second. Thickness is 10 centimeter. And what is the number of planks to stop bullet? To stop bullet, what is the number of planks required? Can you tell me what is the n value here? What is n? N is calculated by u by v for one plank. So n is equal to, what is initial velocity? It is 5. What is final velocity? It is 2.5. So n value is 2, right? N value is 2. So now we shall simplify this. So we can use either the shortcut, n is equal to n square divided by n square minus 1, or we can go for a mathematical step. So we can use this, v square is equal to u square plus 2as, v is 2.5 square and this is 5 square plus 2as. What is s value? S value is 0.1. Calculate the value of 2a. We'll get some value for 2a. Then again use in second equation, v square is equal to 2as, and v is 0, u is 5 square, and you have the value of 2a. Substitute here the value of 2a. And what is the distance traveled? Distance traveled can be written as n into t. T is 0.1. Okay. Or we can use this shortcut. N is equal to, what is n square? N is a 2 square divided by 2 square minus 1. So what is the answer? N is equal to 2 square divided by 2 square minus 1. And this will be 4 by 3, right? What is 4 by 3? 4 by 3 is 1.33. So you have to round up this number, okay? 1.3 is 2. Number of plank required is 2. Why we are rounding up here? Because if n is 1.33, it means there are 2 blocks and the bullet has traveled completely from first block, okay, and after traveling some distance inside the block, for example some 30% it has traveled, then it will comes to rest. So how many planks are required? Total planks required is 2. So if you get a fraction like this, if you get n is equal to 9.2 something, okay, if n is equal to 9.2, how many planks are required? You have to round up this number. 9.2, it means we require 10 planks. Why are getting a fraction here? Sometimes the bullet does not completely travel from here to here. It may stop midway. It will travel completely through first block and it will travel small distance and it comes to rest. So the answer may be in the fraction form, 1.33 or 1.55. So then we can get a fraction, we have to round it up, okay. So try to solve one more problem. There is a block here, it enters with speed 10 meter per second and exits with the speed 2.5. Thickness of the slab is 1 meter. Find number of planks required. Find number of planks required to stop it. Find number of planks required to stop bullet. So here, what is the value of n? N is 4, right? Because n is calculated by u by v. So n is equal to 10 by 2.5. So this is 4. So if you calculate the number of flank, it is n square minus 1 divided by n square. So what is n? This is 4 square minus 1. This is definitely 4 by 3. Sorry. N is 4. So 4 square minus 1 divided by 4 square. N is 4 square is 4 square is 16 minus 1 divided by 16. So some 2 flanks are required here, right? Okay. So sometimes this can be solved in a board exam method also. Okay. This is a small variation here. We can derive shortcut also for that. But the question comes like this: the bullet enters the block with 10 meter per second, and it comes out with the speed of 5 meter. Okay. The bullet will comes out, and they will tell us velocity decreased by 20%. Okay. Velocity decreased by 20%. So what is the number of flanks required? So they will simply give us that the bullet enters the plank by 10 meter per second, and it comes out with the speed of 20%. What is the number of flank? So what we can do here is, if 20% speed decreases, they will give a sentence like speed decreases. The speed decreases by 20%. The speed decreases by 20%. So what happens here? Speed decreases by 20%. What is the number of planks required? So this is a second kind of problem. They will not give us the final velocity. What we have to do is, if initial velocity is 10, or sometimes they may not give this. Okay, they may not give this velocity also. They will just tell that for one plank the speed will decrease by 20%. What is the number of planks required to stop it? So we have to take initial speed as u, and what will be the final speed? So final speed is u minus 20% of u. Okay, and what is this? U minus 20% of u. This can be written as u minus, what is 20%? 20% is always written as 20 by 100 of u. Okay. Then we have to solve this. What is the final velocity? Final velocity is this is 100 minus 20. So this is 80 u by 100. Okay. Or you can take directly. If 20% decreases, what is the remaining part? If 20% decreases, what is the remaining part? Remaining part is 80% of u. So what is 80% of u? It is 80 u by 100. So just remember, what is n% of a? If anyone tells what is n% of a, it is nothing but n by 100 of a. Okay. So I'll draw a question here. You have to solve it. If from one plank, if a velocity decreases by 10%, how many planks are required? How many planks are required to stop bullet? Okay, try to solve this. From one plank, the speed decreases by 10%. How many planks are required? Okay, what we can do about the velocity? So initial velocity is u. Initial velocity is u. What will be the final velocity? First calculate it. So initial velocity, if it is u, then velocity decreases by 10%. So then what is the remaining? It is 90% of u. So if initial velocity is u, what is the final velocity? Final velocity is 90 u by 10, right? Sorry, 90 u by 100. So this is 90% of u is nothing but 0.9 u, right? 0.9 u. Then what we can do here is, we can calculate the value of v square is equal to u square plus 2as. So v square, what is v here? It is 0.9 u whole square. What is u square? It is u square itself. Then what is acceleration? We will write the here acceleration is minus. Then we have 2. What is s here? S is t. So this is 2t. Then what is retardation? It is a. So let us calculate the value of 2ta. So if you solve this, it is 0.9 u square minus u square. It is a value of minus 2ta, right? Then we can use in the second equation, v square is equal to u square plus 2as. Final velocity is 0. What is initial velocity? It is u square. And here this can be written as minus 2a. Distance is nothing but nt. So 0 is equal to u square minus, what is the value of 2at? I already calculated here. Value of 2a and t. I already know the value. It is plus 0.9 u square minus u square into n, right? Then we can solve this. Yes, this is 0.9 nu whole square. This is 0.9 nu whole square, remember it. So let us solve this. U square come here. This is minus u square, and this can be written as u square minus 0.9 square and u square into n, right? And I can take minus outside. So let us do one thing. You take to the left hand side minus u square, and this is 0.9 u whole square minus u square. You can reverse this. If you reverse this, there is a minus here, and we have n. Since I have reversed this, I have to write minus here. So this is u square minus 0.9. 0.9 u square is 0.9 square into u square, right? So this can be taken outside. So minus u square, this is equal to, you can get cancelled with u square. So u square is equal to, this is u square minus 0.9 square is 0.81 u square into n. So we can cancel this u square cancel. Then we are left with 1 minus 0.81 into n, right? Then we can solve this. Okay, some people have doubt here. Why I have taken minus here? Okay, so u square is taken to the left side, it is minus, and here we have 0.9 u square minus u square. I have reversed this. If I reverse this, I can take minus outside, right? I already told you, if a plus minus b is there, I can reverse this. This can be written as b minus a. Only thing I have to do is I have to take minus outside, right? So let us solve this. So what is the answer here? If you solve this, so this will be 1 divided by 0.19. So what is 0.19? 0.19, this will be nearly 5.26, right? So round this up. When you round up 5.26, when you round up 5.26, this is nearly 6, right? So 6 planks are required. How many planks are required? We require 6 planks. So yes, so you have a work now. What you have to do is, if there are problem like this, the block will enter with you and some 10% decreases. What is the final velocity and what is n? You have to calculate this. And if it is 20%, entering speed is u. With what speed does it comes out, and what is the number of planks required? Okay, this can be done in a shortcut also. I will discuss it tomorrow. So this problem can be solved with a shortcut also, but too many shortcuts are difficult to remember. But we will derive it tomorrow. So this whole problem, this can be simplified by using a shortcut. Okay, so I will discuss it tomorrow. So any problem, it can be solved by a shortcut as well as a general method. I have solved it by using a general method. We can use a shortcut to solve it. Okay, there is a direct shortcut to solve it, or we can use a half general method and I can use half shortcut. Okay, but it requires few calculations. There are 2 methods. Other 2 methods are there. I will discuss it tomorrow. Okay, this is time to wind up. So we shall meet tomorrow. Okay. Okay, students. Thank you. So next class will be continued with the same link. Thank you.
Hi everyone, my name is Hevan Patel. I am from Mysore. ನಾನು 10th ನಾನು 65% ತೆಗೆದಿದ್ದೆ. ನಾನು ಈಗ ಎಕ್ಸೆಲ್ಗೆ ಬಂದು ನಾನು 84% ತೆಗೆದಿದ್ದೀನಿ. ಅದಕ್ಕೆ ನಮ್ಮಪ್ಪ ಆ್ಯಕ್ಚುಲಿ ಈಸ್ ಅ ಫಾರ್ಮರ್. ಈಗ ನಾನು ಆ್ಯಕ್ಚುಲಿ ಇಲ್ಲಿ ಬಂದು ಎಕ್ಸೆಲ್ ಬಂದಾದಮೇಲೆ ಎಕ್ಸೆಲ್ ಸೇರ್ಕೊಂಡಿಲ್ಲ. ಇಲ್ಲಿರೋ ಲೆಕ್ಚರರ್ಸು ಇವನ ಸ್ಮಾಲ್ ಡೌಟ್ಸ್ ಇದ್ದರೂ ಎಲ್ಲ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತಾರೆ, ತುಂಬ ಡೀಟೇಲ್ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತಾರೆ. ಎಕ್ಸೆಲ್ ತುಂಬ ನಂಗೆ ಸಪೋರ್ಟ್ ಮಾಡಿದೆ ನನ್ನ ಇಷ್ಟು ಸ್ಕೋರ್ ಮಾಡೋಕ್ಕೆ. ಸೊ ಥ್ಯಾಂಕ್ ಯು ಎಕ್ಸೆಲ್.
ಎಲ್ರಿಗೂ ನಮಸ್ಕಾರ ರೀ. ನನ್ನ ಹೆಸರು ಪ್ರೀತಮ್ ಎಂ ಮಾದಿಗರ್. ನಾನು ಬೆಳಗಾವಿಯಿಂದ ಬಂದೀನಿ. ನಾನು ಎಕ್ಸೆಲ್ಗೆ ಸೇರಿದ ಮೇಲೆ ತುಂಬಾ ನನ್ನಿಂದ ಚೇಂಜಸ್ ಕಂಡ್ಕೊಂಡಿನಿ. ಆ್ಯಕ್ಚುಲಿ ನಾನು 10th 70% ತೆಗೆದಿದ್ದೆ. ಆ್ಯಂಡ್ 10th ಇಲ್ಲಿ ಎಕ್ಸೆಲ್ಗೆ ಸೇರಿದ ಮೇಲೆ ನನಗೆ ಟೀಚರ್ ಸಪೋರ್ಟ್ಸ್ಗಳಿಂದಾಗಲಿ ಅಪ್ಪ ಅಮ್ಮನ ಸಪೋರ್ಟಿಂದಾಗಿ ನಾನು ಇವತ್ತು 91% ತೊಗೊಂಡು ತುಂಬ ಒಳ್ಳೆ ಸ್ಕೋರ್ ಮಾಡಿದ್ದೀನಿ. ನನಗೆ ಅದಕ್ಕೆ ತುಂಬ ಖುಷಿ ಆಗ್ತಿದೆ. ಥ್ಯಾಂಕ್ ಯು ಎಕ್ಸೆಲ್.
ಎಲ್ಲರಿಗೂ ನಮಸ್ತೆ. ನನ್ನ ಹೆಸರು ತೇಜಸ್ DS. ನಾನು ಮೂಲತಃ ಚಾಮರಾಜನಗರ ಜಿಲ್ಲೆಯವನು. ನಾನು ಇಲ್ಲಿಗೆ ಬಂದಮೇಲೆ ತುಂಬ ಚೇಂಜಸ್ ನಾನು ಕಂಡುಕೊಂಡಿದ್ದೀನಿ. ನಂದು 10th 70% ಇತ್ತು, ಆದರೆ ಎಕ್ಸೆಲ್ಗೆ ಸೇರಿದ ಮೇಲೆ ನಂದು 24% ಇನ್ಕ್ರೀಸ್ ಆಗಿದೆ. ಇಲ್ಲಿ ನಾನು 93% ನಾನು ಗಳಿಸಿದ್ದೀನಿ. ತುಂಬ ಖುಷಿಯಾಗಿದೆ. ಮನೇಲಿ ಅಪ್ಪ ಅಮ್ಮ ಹಾಗೂ ನಮ್ಮ ಬಂಧುಗಳೆಲ್ಲ ತುಂಬ ಖುಷಿ ಪಡ್ತಾ ಇದ್ದಾರೆ. ಐ ಥ್ಯಾಂಕ್ ಎಕ್ಸೆಲ್.
ನನ್ನ ಹೆಸರು ಉಲ್ಲಾಸ್ ಮೂಲಿಮನಿ. ನನ್ನ ಊರು ಹುಬ್ಬಳ್ಳಿ. ನಾನು ಈ ಕಾಲೇಜಿಗೆ ಸೇರಿದ ಮೇಲೆ ನನ್ನಲ್ಲಿ ತುಂಬ ಒಳ್ಳೆಯ ಗುಣಗಳನ್ನು ಪಡೆದುಕೊಂಡಿದ್ದೀನಿ. ನಾನು 10th ಇಲ್ಲಿಗೆ ಸೇರುವಾಗ 72% ತೆಗೆದುಕೊಂಡು ಇಲ್ಲಿ ಸೇರಿದ್ದೆ. ಇಲ್ಲಿ ಸುಮನ್ ಸರ್ ಅಥವಾ ನಮ್ಮ ಅಧ್ಯಕ್ಷರು, ನಮ್ಮ ಗುರುಗಳು ನನ್ನನ್ನು ಹುರಿದುಂಬಿಸಿ ಇವಾಗ 92% ತೆಗೆದುಕೊಳ್ಳಲು ಕಾರಣವಾಗಿದ್ದಾರೆ. ಇಲ್ಲಿಯ ಕೋಚಿಂಗ್, ಇಲ್ಲಿಯ ವ್ಯವಸ್ಥೆ ನನಗೆ ತುಂಬ ಹೆಲ್ಪ್ ಆಯಿತು. ಥ್ಯಾಂಕ್ ಯು ಎಕ್ಸೆಲ್. ಥ್ಯಾಂಕ್ ಯು ಸುಮನ್ ಸರ್.
ಎಲ್ಲರೂ ನಮಸ್ಕಾರ. ನಾನು ಮನೋಜ್ ಏಟಿ. ನಾನು ಬಂದು ದಾವಣಗೆರೆಯಿಂದ. ನಾನು 10th 62% ತೊಗೊಂಡಿದ್ದೆ. ಎಕ್ಸೆಲಿಗೆ ಬಂದಮೇಲೆ 88 ತೊಗೊಂಡು ಡಿಸ್ಟಿಂಕ್ಷನಲ್ಲಿ ಪಾಸ್ ಆಗಿದ್ದೀನಿ. ನಾನು ಎಕ್ಸೆಲ್ ಟೀಚರ್ಸ್ಗೆ ತುಂಬ ಥ್ಯಾಂಕ್ಸ್ ಹೇಳೋಕ್ಕೆ ಇಷ್ಟಪಡ್ತೀನಿ. ಥ್ಯಾಂಕ್ ಯು ಎಕ್ಸೆಲ್.
ವೀರೇಶ ಶಶಿಮಠ ಅಂತ. ನಾನು ಗಜೇಂದ್ರಗಢದಿಂದ ಬಂದಿದ್ದೀನಿ, ಗದಗ ಡಿಸ್ಟಿಕ್ಕು. ನಾನು 10th ತುಂಬ ಲೋ ಸ್ಕೋರ್ ಇತ್ತು, 57 ಇತ್ತು. ಆದರೆ ಈಗ ಎಕ್ಸೆಲ್ ಬಂದ ಮೇಲೆ 88% ತೆಗೆದಿದ್ದೀನಿ. ಹಾಗಾಗಿ ನಮ್ಮ ಲೆಕ್ಚರ್ಸ್ಗಾಗಲಿ, ವಾರ್ಡನ್ಸಿಗಾಗಲಿ, ನಮ್ಮ ಚೇರ್ಮನ್ ಸರ್ಗಾಗಲಿ ತುಂಬ ಥ್ಯಾಂಕ್ಸ್ ಹೇಳೋಕ್ಕೆ ಇಷ್ಟಪಡ್ತೀನಿ. ಜೈ ಎಕ್ಸೆಲ್.
ನನ್ನ ಹೆಸರು ತ್ರಿಶೂಲ್ MP. ನಾವು ಶಿವಮೊಗ್ಗ ತಾಲೂಕು ಸೂಗೂರು ಎಂಬ ಗ್ರಾಮದಿಂದ ಬಂದಿದ್ದೀನಿ. ನಮ್ಮಪ್ಪ ವ್ಯವಸಾಯ ಮಾಡ್ತಾರೆ. ನಾನು 10th 27% ಜಾಸ್ತಿ ತೆಗೆದಿದ್ದೀನಿ. 10th 63.2 ತೆಗೆದಿದ್ದೆ. ಇವಾಗ 89.9 ತೆಗೆದಿದ್ದೀನಿ. ಅದಕ್ಕೆ ತುಂಬ ಖುಷಿ ಆಗ್ತಿದೆ. ಧನ್ಯವಾದಗಳು. ಥ್ಯಾಂಕ್ ಯು ಸರ್.
ನನ್ನ ಹೆಸರು ವಂಶಿ ಕೆಂಪಿ. ನಾನು ಅರ್ಕಲ್ಗೂಡು ತಾಲೂಕು ಹಾಸನ ಡಿಸ್ಟಿಕ್ಕಿಂದ ಬಂದಿದ್ದೀನಿ. ನಮ್ಮ ಅಪ್ಪ KSRTC ಕಂಡಕ್ಟರ್. ನಾನು 10th ತುಂಬ ಲೋ ಸ್ಕೋರ್ ಮಾಡಿದ್ದೆ. ನನ್ನ ಪರ್ಸೆಂಟೇಜ್ 58 ಇತ್ತು. ಇಲ್ಲಿ ಎಕ್ಸೆಲ್ಗೆ ಬಂದಮೇಲೆ ನನ್ನ ಪರ್ಸೆಂಟೇಜ್ 93 ಆಗಿದೆ. ಅದಕ್ಕೆ ನಾನು ನಮ್ಮ ಎಕ್ಸೆಲ್ ಸಂಸ್ಥೆಗೆ ಥ್ಯಾಂಕ್ಸ್ ಹೇಳೋಕ್ಕೆ ಇಷ್ಟಪಡ್ತೀನಿ. ಥ್ಯಾಂಕ್ ಯು ಎಕ್ಸೆಲ್.
ನಮಸ್ತೆ, ನನ್ನ ಹೆಸರು ಶ್ರೇಯಸ್. ನಾನು ತುಮಕೂರು ಡಿಸ್ಟಿಕ್ಕಿಂದ ಬಂದಿದ್ದೆ. ನಮ್ಮ ಪೇರೆಂಟ್ಸ್ ಬಿಸ್ನೆಸ್ ಮಾಡಿದ್ದಾರೆ. ನಂದು 10th ಮಾರ್ಕ್ಸ್ 66.8. ಇಲ್ಲಿ ಎಕ್ಸೆಲ್ ಬಂದಮೇಲೆ ನನ್ನ ಸೆಕೆಂಡ್ PU ಮಾರ್ಕ್ಸ್ 93. ಈ ಎಕ್ಸೆಲ್ ತುಂಬ ಹೆಲ್ಪ್ ಮಾಡಿದೆ. ಥ್ಯಾಂಕ್ ಯು ಎಕ್ಸೆಲ್.
ನನ್ನ ಹೆಸರು ಉದಯಗೌಡ SS. ನಾನು ಮಂಡ್ಯ ಡಿಸ್ಟಿಕ್ ನಾಗಮಂಗಲ ತಾಲೂಕಿಂದ ಬಂದಿರೋದು. ನಮ್ಮಪ್ಪ LIC ಅಡ್ವೈಸರ್, ನಮ್ಮಮ್ಮ KV ರೆಸ್ಕಾಮ್ ಓವರ್ಸಿಯರ್. ನಂದು 10th 67% ಇತ್ತು. ಈಗ ಸೆಕೆಂಡ್ PU 91% ಇದೆ. ಆದರೆ ಥ್ಯಾಂಕ್ ಯು ಫಾರ್ ಎಕ್ಸೆಲ್.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನನ್ನ ಹೆಸರು HM ಹಿತೇಶ್ ಚಂದನ್. ನಾನು ನಂಜನಗೂಡು ತಾಲೂಕು ಮೈಸೂರು ಜಿಲ್ಲೆಯಿಂದ ಬಂದಿದ್ದೇನೆ. ನಾನು ಫಸ್ಟು 10th ಸ್ಟ್ಯಾಂಡರ್ಡ್ ನಂಜನಗೂಡಲ್ಲಿ ಓದಿದ್ದೆ. ನನಗೆ ಪರ್ಸೆಂಟೇಜ್ 61 ಬಂದಿತ್ತು. ಈ ಎಕ್ಸೆಲ್ ಸಂಸ್ಥೆಗೆ ಬಂದ ಮೇಲೆ ನನ್ನ ಪರ್ಸೆಂಟೇಜ್ ಸೆಕೆಂಡ್ PUC 91% ಬಂದಿದೆ. ಇದಕ್ಕೆಲ್ಲ ಕಾರಣ ನಮ್ಮ ಅಧ್ಯಕ್ಷರಾದ ಸುಮಂತ್ ಕುಮಾರ್ ಜೈನ್ ಮತ್ತು ಸಂಸ್ಥೆ. ಥ್ಯಾಂಕ್ ಯು ಫಾರ್ ಎಕ್ಸೆಲ್.
ಶಶಾಂಕ್ ಶಾನ್ಭೋಗ್. ನಾನು ಚಾಮರಾಜನಗರ ಡಿಸ್ಟಿಕ್ ಗುಂಡ್ಲುಪೇಟೆ ತಾಲೂಕು. ನಾನು ಸ್ಟಾರ್ಟಿಂಗ್ ಎಕ್ಸೆಲ್ಗೆ ಬಂದಾಗ 56% ಇತ್ತು 10th. ಅದರ ಸೆಕೆಂಡ್ PUC 86% ಬಂದಿದೆ. ಲೆಕ್ಚರರ್ಸ್ ತುಂಬ ಸಪೋರ್ಟ್ ಮಾಡಿದೆ. ಥ್ಯಾಂಕ್ ಯು ಎಕ್ಸೆಲ್.
Hello everyone, I am Shreya Shukre from Bidar. My father is Deputy Secretary and In my 10th standard, I have just scored 62% because of my carelessness, and I have increased my 28% by scoring 91% in the second PU exam. I am really thankful to Mr. Suman Kumar Jain, who have provided me this opportunity to enter into this college and score well. I am really thankful to the faculties of this college. Thank you, Excel.
Bhuvanesh, I am from Bengaluru. At my 10th, I have 64%. Now I increased 30%, 95%. And thank you for all my faculties who have taught me, and thank you, Excel.
ಹಲೋ ಎವ್ರಿ ಒನ್, ನನ್ನ ಹೆಸರು ಲಿಖಿಜಿ ಪುಟಾಣ್ಕರ್. ನಾನು ಐ ಆಮ್ ಫ್ರಮ್ ಚಿಕ್ಕಮಗಳೂರು. ನಾನು ಟೆಂತಲ್ಲಿರ್ಬೇಕಾದ್ರೆ ನಂದು 54% ಇತ್ತು. ಆದರೆ ಇಲ್ಲಿ ಎಕ್ಸೆಲಿಗೆ ಬಂದಮೇಲೆ ನನಗೆ 88.4 ಬಂದಿದೆ. ನನಗೆ ಇಲ್ಲಿಂದ ಎಕ್ಸೆಲಿಂದ ಸಿಕ್ಕಾಪಟ್ಟೆ ಸಪೋರ್ಟ್ ಸಿಕ್ಕಿದೆ. ಥ್ಯಾಂಕ್ ಯು ಎಕ್ಸೆಲ್.
ಹಲೋ ಎವ್ರಿವನ್, ನನ್ನ ಹೆಸರು ವಿಶಾಲ್ ಅಂತ. ನಾನು ಗುಲ್ಬರ್ಗಾಯಿಂದ ಬಂದಿದ್ದೀನಿ. ಎಕ್ಸೆಲ್ ಪಿ ಯು ಕಾಲೇಜಿಗೆ ಸೇರೋಕ್ಕಿಂತ ಮುಂಚೆ ನಂದು 10th 66% ಇತ್ತು. ಅದಕ್ಕೆ ಈ ಎಕ್ಸೆಲಲ್ಲಿ ಸೇರಿದಾಗ ಸೆಕೆಂಡ್ ಪಿ ಯುಲಿ ನಾನು 94% ತೆಗೆದಿದ್ದೀನಿ. ಸುಮಾರು 28% ಇನ್ಕ್ರೀಸ್ ಆಗಿದೆ ನನಗೆ. ಇದಕ್ಕೆ ಸಪೋರ್ಟ್ ಮಾಡಿರುವ ಎಕ್ಸೆಲ್ ಪಿ ಯು ಕಾಲೇಜಿನ ಗುರುಗಳು, ಶಿಕ್ಷಕರು ಎಲ್ಲರಿಗೂ ಧನ್ಯವಾದ ಹೇಳುತ್ತಾ, ನಮ್ಮ ಕಾಲೇಜಿನ ಅಧ್ಯಕ್ಷರಾಗಿರೋ ಸುಮಂತ್ ಕುಮಾರ್ ಸರಿಗೆ ತುಂಬ ಹೃದಯದ ಧನ್ಯವಾದ ಹೇಳ್ತೀನಿ. ಥ್ಯಾಂಕ್ ಯು ಎಕ್ಸೆಲ್.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನನ್ನ ಹೆಸರು ಶ್ರೇಯಸ್ ಹಿರೇಮಠ್. ನಾನು ಬೆಳಗಾವಿ ಜಿಲ್ಲೆಯ ಮೂಡಲಗಿ ತಾಲೂಕಿನಿಂದ ಬಂದಿದ್ದೀನಿ. ನಂದು 10th 53% ಇದ್ದು. ಆದರೆ ಇವಾಗ ಸೆಕೆಂಡ್ ಪಿ ಯು ಸಿಯಲ್ಲಿ 91% ಆಗಿದೆ. 37% ಇನ್ಕ್ರೀಸ್ ಆಗಿದೆ. ಇದಕ್ಕೆಲ್ಲ ಕಾರಣ ನನ್ನ ಹಾರ್ಡ್ ವರ್ಕ್ ಮತ್ತು ಅಪ್ಪ ಅಮ್ಮ ಮತ್ತು ಎಕ್ಸೆಲ್ ಕಾಲೇಜ್ ತುಂಬ ಸಪೋರ್ಟ್ ಮಾಡಿದ್ದಾರೆ. ಸೊ ಥ್ಯಾಂಕ್ ಯು ಎಕ್ಸೆಲ್.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನನ್ನ ಹೆಸರು ಸಂಜಯ್ ಕುಮಾರ್. ನನ್ನ ಊರು ದಾವಣಗೆರೆ ಡಿಸ್ಟಿಕ್, ಹೊನ್ನಾಳಿ ತಾಲೂಕು. ನನ್ನ ತಂದೆ ಎಕ್ಸ್ ಆರ್ಮಿ, ಈಗ ಕೆನರಾ ಬ್ಯಾಂಕಲ್ಲಿ ಉದ್ಯೋಗ ಮಾಡ್ತಿದ್ದಾರೆ. ಮತ್ತು ನಾನು 10th ಕಡಿಮೆ ಅಂಕಗಳನ್ನು ಗಳಿಸಿದ್ದೆ. ಈಗ ಎಕ್ಸೆಲ್ಗೆ ಬಂದಾದ ಮೇಲೆ ನಾನು ಈಗ ಎಕ್ಸೆಲ್ ರಿಸಲ್ಟ್ ಹೇಳಲಿಕ್ಕೆ ಇಷ್ಟಪಡ್ತೀನಿ. ನಂದು 95% ನಾನು ಎಕ್ಸೆಲ್ ಮತ್ತು ನನ್ನ ಗುರುಗಳು ಮತ್ತು ನನ್ನ ತಂದೆ ತಾಯಿ ಆಶೀರ್ವಾದದಿಂದ ನನಗೆ ಒಳ್ಳೆಯ ಅಂಕಗಳನ್ನು ಗಳಿಸಿದ್ದೇನೆ. ಇದು ಎಕ್ಸೆಲ್ ಹೆಸರು ಮಾತ್ರವಲ್ಲ, ಇದೊಂದು ಬ್ರ್ಯಾಂಡ್ ಅಂತ ಹೇಳಲಿಕ್ಕೆ ಇಷ್ಟಪಡ್ತೀನಿ. ಧನ್ಯವಾದಗಳು. ಥ್ಯಾಂಕ್ ಯು ಎಕ್ಸೆಲ್.
ನಮಸ್ಕಾರ ಎಲ್ಲರಿಗೂ. ನನ್ನ ಹೆಸರು ಸಂದೀಪ್ ಹುಟ್ಗಿ. ನಾನು ಬಿಜಾಪುರ ಜಿಲ್ಲೆಯಿಂದ ಬಂದಿದ್ದೇನೆ. ನಮ್ಮ ತಂದೆ ಹೆಸರು ಪ್ರಕಾಶ್ ಹುಟ್ಗಿ. ಅವರು ರೈತರಿದ್ದಾರೆ. ನಾನು 10th 74% ಮಾಡಿದೆ. ಆಗ ನನಗೆ ಕಾನ್ಫಿಡೆನ್ಸ್ ಇರಲಿಲ್ಲ ಸೈನ್ಸ್ ಸ್ಟ್ರೀಮ್ ತೊಗೋಕ್ಕೆ. ಆದರೆ ನಮ್ಮಪ್ಪ ಎಕ್ಸೆಲ್ ಕಾಲೇಜ್ಗೆ ಸೇರಿಸಿದ್ರು. ಅದರಿಂದ ನನಗೆ ಸೆಕೆಂಡ್ ಪಿ ಯುಯಲ್ಲಿ 96% ಬಂದಿದೆ. ಇದಕ್ಕೆಲ್ಲ ಕಾರಣ ನಮ್ಮ ತಂದೆ ತಾಯಿ ಸಪೋರ್ಟಿನಿಂದ ಮತ್ತು ಎಕ್ಸೆಲ್ ಸಪೋರ್ಟಿನಿಂದ. ಎಕ್ಸೆಲ್ ತುಂಬ ಒಳ್ಳೆಯ ಕಾಲೇಜ್ ಆಗಿದೆ. ಥ್ಯಾಂಕ್ ಯು ಎಕ್ಸೆಲ್.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನನ್ನ ಹೆಸರು ಹೇಮಂತ್ ಜಿ ಎಸ್.
Good morning, dear students. Hope you're all doing well, and hope you all had your breakfast and also attended the last class as well. So, welcome back to the botany class. We were discussing regarding how new cells are getting formed. What is the process of cell division? Cell division ಅನ್ನೋದು ಹೇಗಾಗುತ್ತೆ? What is that? ಅನ್ನೋದ್ರ ಬಗ್ಗೆ we were discussing. As you all know, in cell cycle we have two phases. In cell cycle we have two phases. One is interphase, another one is the M phase. Okay, the interphase and the M phase. In interphase we have three subdivisions basically: G1, S and G2. G1, S and G2. In G1 phase, what happens basically? Cell growth takes place. Many organelles divide. New organelles get formed. Okay. And in S phase, what takes place? S phase is also referred as a synthesis phase. It is also referred as synthesis phase, where the DNA replication takes place. DNA replication takes place. In which the chromosome number do not change. The chromosome number do not change. But the content of DNA doubles. It gets doubled. Content of DNA double agate, but the chromosome number will not change. Okay, will not change. If you see, let us solve something here. let this be the content of DNA and this is the chromosome number. During G1 phase and S phase, what happens annodana we shall see. Okay. Now you have to answer to this question. You have to answer to this question. If the content of DNA during G1 phase is 4C, if content of DNA during G1 phase is 4C, if the chromosome number is 2, what happens to the content of DNA and chromosome number in S phase? I want the answer for that. What happens for the content of DNA and the chromosome number in S phase? I want everyone to answer this. Only then, after most of you have answered, only then I'll tell the actual answer. Yes, most of you have answered this. Correct. The content of DNA gets doubled in S phase. That becomes 8C, but the chromosome number remains same. It remains as 2 only. Very good. Okay, that's regarding the S phase. Sir, 4C will double and become 8C and 2C, 2 will make same. Perfect. Okay, and there is one more extra process that takes place in, the animal cell. Okay, animal cell alone the extra process agutte. What is that? In animal cell, I'll write it here. In animal cell, during S phase, why it doubles? See, the doubling takes place because in last class I had given an example. With an example we had discussed. You have a house, okay, you have a house in which you have a single sofa, a single TV, a single fridge and a single washing machine. If you want to divide that into 2, if you want to divide that into 2, can you divide that TV, like can you break that TV into half and divide it to 2? Will that be useful? No, right? In the same way, cell division andag ಏನು ಹೇಳ್ತೀವಿ, we say that from single cell 2 new cells are getting formed. So what and all is there in this cell? What contents are there in this cell? The same contents should be present in the other cell as well. Okay, the same content should be present in the other cell as well. Right? ಅದರಲ್ಲೂ same content ಇರಬೇಕು. ಹಾಗಿರುವಾಗ if 1 gram of DNA is in this, 1 picograms generally, if 1 picograms of DNA is there in this, then in each cell there should be 1 picogram as well. Right? ಎರಡೂ ಸೆಲ್ ಅಲ್ಲೂ ಕೂಡ 1 ಪಿಕೋಗ್ರಾಮ್ಸ್ ಇರಬೇಕು. 0.5 ಪಿಕೋಗ್ರಾಮ್ಸ್ ಹಾಕಿದ್ರೆ ಆಗುತ್ತಾ?
Will that be correct? No. Why? In the next cell, there will be 0.25. In the next cell that divides from this, it goes on reducing. Am I right? So, in order to maintain this number, the DNA gets doubled. Not only the DNA, not only the DNA, all the organelles gets doubled. All the organelles gets doubled, which takes place in G1 phase or G2 phase. G1 phase or G2 phase. ಅಲ್ಲಿ ಬೇಕಾಗಿರುವಂಥವು ಏನಾಗುತ್ತೆ ಡಬಲ್ ಆಗ್ತಾ ಹೋಗ್ತವೆ. Okay, is this clear? And what is that process? The process is known as the replication of DNA. Here it is there. DNA replication is the process. Regarding the different steps of DNA replication, you are going to study that in the second PU or 12th. Hope your doubt is clarified. Kumuda and Neeta Chopade. Yes or no? Do you have any other doubt in this? See, the DNA replication, I cannot explain it now because it is quite a complex topic. It comes in your second PU, okay? So, if I explain it now, it will be difficult, or I can give just an introduction on that. Generally, you know that the DNA is a DNA is a double helical structure, right? A DNA is a biopolymer that has a double helix like structure. It is a double stranded structure. From this double stranded structure, the two strands gets separated, in which a new strand will be added, This is in simple what DNA replication is, but the process is not as simple as that I have told now. Okay. If I go in depth, it will be a bit difficult for you to understand it now, because there are many enzymes that take part. You should know about the DNA structure as well. DNA is not as simple as I have written here. It may look simple, but it isn't. The chemicals that are present in DNA, it is quite complex and all. Okay. Now we got to know what a DNA replication is, or you. Simple definition, DNA replication is nothing but formation of, it is a process of a formation of new copy of DNA. Okay. Or Xerox, DNA na Xerox tegidre ಒಂದು extra Xerox ಬರುತ್ತಲ್ಲ. See, the same DNA will be formed here in 2 copies. One DNA ಇದ್ದು 2 copies ಆಗಿ ಫಾರ್ಮ್ ಆಗುತ್ತೆ. Okay, one DNA ಇದ್ದು 2 copies. Xerox ತೆಗೆಯೋ ತರ. It is like you're taking Xerox. Did you get it? It is nothing but formation of new copy of a DNA. Photocopying a DNA. DNA Xerox ತೆಗೆದ್ರೆ 2 ಫೋಟೋ ಕಾಪೀಸ್ ಸಿಗುತ್ತಲ್ಲ, ಅದನ್ನ DNA ರೆಪ್ಲಿಕೇಶನ್ ಅಂತ ಕರೀತೀವಿ. At the beginning of cell division, G1 phase ಅಲ್ಲಿ, the DNA content will be this much ಅಂತ ಅನ್ಕೊಳ್ಳಿ. Okay, it gets doubled there. It gets doubled. Contents ಜಾಸ್ತಿ ಆಗುತ್ತೆ. Why? Why? In order to distribute them into two cells. In order to distribute them into two cells. ಅದನ್ನು ಟೂ ಸೆಲ್ಸ್ಗೆ ಈಸಿಯಾಗಿ ಡಿಸ್ಟ್ರಿಬ್ಯೂಟ್ ಮಾಡಲಿಕ್ಕೋಸ್ಕರ ದಿಸ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್. Okay. Okay. Yes, hope everything regarding the DNA replication is clear. So in S phase, there is one more extra process that takes place in. Sir, father having one disease, son also that diseases cannot remove. You're asking that if father is also having a disease, it gets transferred to a son. There are some diseases that get transferred to the next generation as well. Some do not transfer. Okay, some do not transfer. If they are linked on to the DNA ಗೆ linked ಆಗಿರುವಂತ disease ಆಗಿದ್ರೆ, it gets transferred to the next generation. ಅದರಲ್ಲೂ there are many aspects. ಅದು ನೀವು ಸೆಕೆಂಡ್ ಇಯರ್ ಅಲ್ಲಿ ಕರೀತೀರ, ಡಿಸೀಸ್ ಹೇಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗುತ್ತೆ, ಯಾವ ಡಿಸೀಸ್ಗಳು ಟ್ರಾನ್ಸ್ಫರ್ ಆಗುತ್ತೆ ಅನ್ನೋದ್ರ ಬಗ್ಗೆ. Okay, yeah. So in animal cell, there is one more extra process that takes place. Our extra process ಏನು ಅಂತ ಹೇಳಿದ್ರೆ, centriole duplication takes place. How the DNA got doubled in the same way, there is an organelle called as a centriole. Centriole ಅಂತ ಒಂದು organelle ಇದೆ, which will be somewhat like this, whose structure will be somewhat like this. This is a centriole. ಇದನ್ನ centriole ಅಂತ ಕರಿತೀವಿ ನಾವು, which is very much required for cell division. It is required for cell division, and it is only found in animal cell. So centriole duplication also takes place in animal cell. It will not take place in plant cell because centriole is not found in plant cell. Centriole illa plant cell alli, as they have centriole in animal cell. In animal cell, along with the DNA replication during S phase, the centriole also duplicates. Function I have mentioned already, cell division. It helps in cell division. When the process of cell division comes, you will get to know how centriole acts there. Okay. And the centriole duplication, it takes place in cytoplasm. Whereas the DNA replication takes place in nucleus. DNA replication takes place in nucleus. Centriole duplication takes place in cytoplasm. Is this clear? Yes? Okay then. Let us move on to the G2 phase. See, regarding the beginning of college, you will get notified. Okay, notified. You can receive your uniforms now also. You can receive your uniforms now as well. You can visit to the college, or you can talk to the office regarding any other alternative methods. Okay. Yes. On to the last stage in interface, that is G2 phase. G2 phase. Here what happens? In G2 phase, here also the cell growth continues. That means other organelles, they also undergo multiplication, along with which proteins required for cell division are synthesized. Okay, the proteins which are required for the cell division are synthesized. If you have any doubts regarding those uniform and all, you can call to the concerned office or the one who is in touch with you. Okay, ನಿಮ್ಮ ಹತ್ರ admission team ಅವರು ಯಾರೋ ಒಬ್ರು ಟಚ್ ಅಲ್ಲಿ ಇರ್ತಾರಲ್ಲ, you can call to them and clarify any doubts regarding that. Okay, so G2 phase, the cell growth continues and the proteins required for cell division gets synthesized. Some proteins will be required during cell division. Those proteins gets synthesized during which phase? G2 phase. Are you clear about G1, yes, and G2? If you have any doubt, you can ask. The G2 phase is the last phase of interface. G2 phase is the last phase of interface. It is not the last phase of cell division. Yes, after the G2 phase, the cell divides. The actual dividing phase begins, known as M phase. Okay, the actual dividing phase begins, which is known as M phase, that takes place after the G2. Illi tanaka, till G2 phase, the cell prepared itself for division. Now the preparation is completed, it will enter into dividing phase. Before discussing about that, what is the time period taken by the cell for performing, or what is the time period took by the cell in interface, and what will be the time taken by or taken by the cell in the M phase? What would be the time taken by the cell in interface, and what will be the time that the cell will be taking in M phase? M phase ಅಲ್ಲಿ ತಗೊಳ್ಳುವಂತ ಟೈಮ್ ಎಷ್ಟಿರ್ಬೋದು, ಇಂಟರ್ಫೇಸ್ ಅಲ್ಲಿ ತಗೊಳ್ಳುವಂತ ಟೈಮ್ ಎಷ್ಟಿರ್ಬೋದು? Anybody? Can you guess? Is it 50-50? Yes, M means mitosis only. Interphase is not the last. Interphase is the first phase. Followed by interphase is the M phase. Under interphase, G2 is the last phase. Yes, what would be the time taken by the cell in interphase and M phase? Anybody? You can guess. You can roughly guess, okay? Even if you have made given wrong answer, it is not a problem at all. M means not metaphase. M refers to mitotic phase or mitosis. The time taken under interphase or in interphase is 95%. If a cell cycle is of about 24 hours, if it is of about 24 hours, then under 24 hours, most of the time will be taken by the interphase. Okay, 23 hours varage the cell will be in interphase. Whereas the M phase, it is just about 5%. For example, there is a bacteria known as E. coli, which divides in about 20 minutes. It divides in about 20 minutes, in which the interphase takes place about 18 minutes, and the 2 minutes is just 2 minutes is the M phase. So in a cell cycle, the 95% of the cell time is taken in interphase, and only 5% of time is taken by the cell in M phase. There is a life lesson in this that everyone should learn. ಎಲ್ಲರೂ ಕಲ್ತ್ಕೋಬೇಕಾದಂತ ಒಂದು ಲೈಫ್ ಲೆಸನ್ ಇದೆ. What is that? What did I mention while discussing about interphase? It is a preparatory phase where the cell is preparing for division. What is the time taken for preparation? The time taken for preparation is 95% of time. That means 18 minutes if the division is of 20 minutes. And for execution, it just took 5%. Okay, 5% of the time. So one should understand that if you prepare very well, if your preparation time is more, then easily the execution will take place. ಕರೆಕ್ಟ್ ಆಗಿ ಪ್ರಿಪರೇಷನ್ಗೆ ಅಂತ ನೀವು ಜಾಸ್ತಿ ಟೈಮ್ ಕೊಟ್ಟರೆ ನಿಮ್ಮ ಎಕ್ಸಿಕ್ಯೂಷನ್ ಏನಿದೆ, ಯಾವುದೇ ಒಂದು ಯಾವ ಪ್ರೊಸೆಸ್ನ ನೀವು ಮಾಡ್ಲಿಕ್ಕೆ ಹೋಗ್ತೀರಾ, that will go on very easily, ಬೇಗ ಆಗುತ್ತೆ. If you don't give enough time for the preparation, then the execution will be very late. Okay, so this is regarding the interphase. interface. Do you have any doubt in this? Now let us go further in this calculation only. Okay, I'll give you some calculations. We need to make a lot of preparation in a single job. Yeah. Okay. I'll give some calculation for you. Now we have added one more stage there. Okay, let's see how many of you will answer quickly to this. This is the content of DNA and this is the chromosome number. This is G1, yes, G2, G1, yes, G2, solve these three. Okay, anybody got the answers for these? Anyone? Very easy. You know the initial amount of the content of DNA as well as the chromosome number that we have. You just have to write the content of DNA in S and G2. Content of DNA as well as chromosome number. I want everyone to answer this. Everyone to answer this. Later I'll move on to, or I'll tell the answer. Okay, final again, non answer. Try. Very easy. You just apply what we had discussed and what changes take place in which stage. If you know that, then it will be very easy. Okay, some are attempting, very good. At which stage is that? You have written 32C, 14C, 14, 56C, 14. In which stage? I want you to write in that order. Like in G1 phase, this is the content. In G2, this is the content. In GSC, this is the content. Okay, I have received only 2 members answer. Okay, let's solve this now. See, in S phase what happens? The content of DNA gets doubled, right? The content of DNA gets doubled. The initial content is 8C. The initial content is 8C. What will it become? It becomes 16C. It becomes 16C. But the chromosome number remains same. The chromosome number remains same. It is 4 only. Right? In G2, was there any difference in DNA? DNA divide ಆಯ್ತಾ or multiply ಆಯ್ತಾ? ಏನಾದರೂ difference ಆಯ್ತಾ? No difference, no change. So the 16C remains the same. The 16C remains the same. The chromosome number remains the same. Perfect, right? Perfect, right? Is that clear? Is that clear? Okay, so in chromosome number there is no change, but in content of DNA the content gets doubled. But in G2 there is no change. In G2 there is no change. Coming on to the second one, 16C becomes what? 32C. In G2 it becomes 32C. Chromosome number, it will remain same, 14 and 14. Okay, on to the next one now. 28. What is the next content or content of DNA in the S phase? It becomes 56C. The chromosome number, it will remain same. Hope this is clear. If you have any doubt in this, you can ask. Is that clear? Yes. Okay. Yes. Now on to the actual dividing phase. Which is that actual dividing phase? It is the M phase. The M phase can also be called as the mitotic phase. It can also be called as the mitotic phase or the actual dividing phase. The actual dividing phase. So there are basically two stages under M phase. Okay, there are basically two stages under M phase. The two stages are karyokinesis and cytokinesis. Okay, karyokinesis and cytokinesis. Let's break this word into two. Karyon and kinesis. Cyto and kinesis. The kinesis refers to division or breaking. Okay. What do you mean by karyon? We have discussed that as well. Prokaryotic cell, eukaryotic cell, in which the karyon refer to nucleus. It refers to nucleus. So, what do you mean by karyokinesis? It is the division of nucleus. The nuclear division is known as karyokinesis. Then what is meant by cytokinesis? It is the division of cytoplasm or the cell division. Okay, the karyokinesis and cytokinesis. For our better understanding, we have again classified karyokinesis into 4 substages. This actually do not exist in nature, but we have divided that into 4 substages for our understanding. Okay, for our understanding, we have subdivided them. What are those 4 substages? The 4 substages are prophase, metaphase, anaphase and telophase. Okay, the 4 substages of karyokinesis are prophase, metaphase, anaphase and telophase. What are those? See, generally what happens is that who named it, a cell used to divide into 2, right? In some intervals he used to observe. So in one interval he could see some change in the cell. In another interval he could see some changes, in another. So he named these into prophase, metaphase, anaphase, telophase, in which major changes could be seen. You asked, right, what are those? So major changes, nucleus alli ಏನು major change ಆಗ್ತಿತ್ತು. What were the major changes that were taking place in the nucleus during the karyokinesis? He used to observe them in different intervals. Okay, 4 interval observe ಮಾಡಿದಾಗ in 4 intervals he could see some of the major changes in them. So he named those stages as prophase, metaphase, anaphase and telophase. Is that clear? Do you have any doubt in this? Yeah, major changes in nucleus ಏನೆಲ್ಲ ಆಗ್ತಾ ಇತ್ತು, those he named it as a stage because it is not easy to study on a whole. Karyokinesis na direct ಆಗಿ ಒಂದೇ stage ಆಗಿ you cannot study because it is a long process. Okay, as it is a long process, you cannot study them in single step. Single step study ಮಾಡಿದ್ರೆ, see here, there will be one nucleus. It will be divided into 2 nucleus. ಅಷ್ಟೇ ಆಗುತ್ತೆ. We will not get to know how this single divide is getting single nucleus is getting divided into two. Okay, ಒಂದು ಇಷ್ಟು ದೊಡ್ಡ ಮಣ್ಣ ತರ ಇದ್ದಿದ್ದು ಅದು ಒಂದು ಥ್ರೆಡ್ ತರ ಇದ್ದಿದ್ದು ಹೇಗಾಗುತ್ತೆ. See, the function is not there. These are the stages where what happens. We will be discussing about that individually. Prophasally, what changes takes place in a cell. Metaphasic bandaga, what changes can be observed. Anaphasic cell entradaga, what process takes place. Telophasic bandaga, what process takes place. In prophase, chromosomes move away and the nucleus dissolve. Chromosomes doesn't move away. The conversion of chromatin network into chromosome take place. Okay, there is no function for each step. These are the steps of a process. Okay, ಒಂದು whole process. ಒಂದು ನ್ಯೂಕ್ಲಿಯಸ್ ಇದ್ದಿದ್ದು 2 cells ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗುವಾಗ ವಾಟ್ ಸ್ಟೆಪ್ಸ್ ಇಟ್ ಅಂಡರ್ಗೋಸ್ ಅದನ್ನ ಪ್ರೊಫೇಸ್, ಮೆಟಾಫೇಸ್, ಅನಾಫೇಸ್ ಅಂಡ್ ಟೀಲೋಫೇಸ್ ಅಂತ ಕರಿತೀವಿ. Okay, this is not PMAT. I have just written its name in capitals ಅಷ್ಟೇ. ಒಂದು ಸ್ಮಾಲ್ ಲೆಟರ್ಸ್ ಅಲ್ಲಿ ನೇಮ್ ಮಾಡಿದ್ದೀನಿ. ಪ್ರೊಫೇಸ್ ಅಂದ್ರೆ ಫುಲ್ ಬರೆಯೋದಕ್ಕೆಲ್ಲ ಜಾಗ ಇರಲಿಲ್ಲ. ದಟ್ ಇಸ್ ವೈ P refers to prophase, M refers to metaphase, A refers to anaphase and T refers to telophase. Okay, so what is M phase basically? M phase is the actual dividing phase, also referred as mitotic phase. There are two stages. The two stages are karyokinesis and cytokinesis. Karyokinesis is nothing but a division of nucleus. Cytokinesis is nothing but a division of cytoplasm. Okay, is that clear? Shall we look on to these stages individually? ಸೊ ಒಂದು ಸೆಲ್ ಇದ್ದಿದ್ದು ಇನ್ನೆರಡು ಹೊಸ ಸೆಲ್ ಆಗಿ ಹೇಗೆ ಫಾರ್ಮ್ ಆಗುತ್ತೆ ಅಂತ ನೋಡೋಣ ಫಸ್ಟಿಗೆ. Okay, there will be a single cell. Within the cell, what do we have? We will have a long nucleus. A nucleus will be there. A nucleus will be there. And the vacuole will be there. Correct? Vacuole will be there. And other organelles will be there as well. Mitochondria, chloroplast, etc., etc. if it is a plant cell. Okay, so this single cell gets divided into two cells. Okay, it gets divided into two cells. Which will be similar to the already existing cell. Now this cell may be called as a parental cell. Yeah, it was there in your 10th as well. 10th ಅಲ್ಲಿ ಇತ್ತು ನಿಮಗೆ cell cycle, cell division, cell cycle ಇರಲಿಲ್ಲ. As far as I remember, you had cell division. Okay, now this can be called as a parental cell. Okay, and these two can be called as daughter cells. These two can be called as daughter cells. See, that is why there is a single cell. There is only one nucleus. There is only one vacuole. There is only one mitochondria and all. But the cells that are getting formed after a division are two. And both the cells should contain one nucleus. Both of them should contain one mitochondria. Okay, so how this take place anodana we shall see in the next class. Okay, hope everything regarding today's class is clear. If you have any doubt, you can ask in the next class as well. Thank you so much for listening to the class. We shall meet in the next class. Thank you.